Hello and good afternoon, everyone. Welcome to day 4 of 5 of the Western Regional Panel on Aquatic Nuisance Species annual meeting. We are so glad that you're here today. Thank you for taking time to join us. We have an outstanding session lined up for you today. For those of you that don't know me, my name is Elizabeth Brown. I'm the former chair of the Western Regional Panel, and I'm currently serving as an advisor to the Executive Committee. I just want to give a shout out to all of our executive committee members. You are outstanding. Thank you so much for your service. And I especially want to say thanks to Dennis Zabaglo, Martha Volkoff, and Mason Parker for just all their hard work. Thank you so much. Okay, a few announcements before we get into today's session. We do have a social space up on Facebook. We'd love for you to join us. We're having a lot of fun interactions in there. And you can get that link on our annual meeting webpage on westernregionalpanel.org. We also have a drawing from one of our sponsors, Astonish, and so you can get that information in the Facebook group and again on our webpage. And so please participate in the drawing and you can win a fun prize. And then finally, our voting membership. You have an election vote ballot in your email. Today is the last day to vote, so please take a minute if you haven't already and vote for new executive committee members and members and approval of old minutes and things of that nature. Today's the last day, so get that done today for us so that we have all of that tallied for tomorrow's business meeting. Okay, as far as this presentation goes, please, we'd love to have your questions. We're going to hold all the questions until the end of the presentations, and then we have about 30 minutes for Q&A that will be led by our coordinator, Mason Parker. So please enter your questions into the Q&A and know that we are logging them and we will answer them at the end after the presentations. And if there's anything we don't get to, we'll have our speakers follow up, okay? And yeah, with that, I'm going to get right to it. So like I said, we have an absolutely outstanding agenda for you today, and I'm so excited to be able to be here with you moderating. Our first presentation is a powerhouse of professionals here in our community. We have Jeffrey Morissette from the Department of Interior, Adam Sepulveda from the U.S. Geological Survey, who also chairs our eDNA committee or work group, Yael Pasamenik from the U.S. Bureau of Reclamation, who is our former lab work group chair and a co-leader on our eDNA committee, and Staj Burgel from the National Invasive Species Council. So with that, I'm going to turn it over to this outstanding group. Thank you for being here today, and I'm going to mute myself out and let you take it over.
Hello, everyone. Let's pull up the slides.
I'm seeing presentation mode, so you might just want to switch your display settings.
Is that better?
Yes, perfect.
Okay, little delay in selecting the monitors. This is Jeff Morissette, and I'm running this slide, but I'm going to quickly pass it over to Stas, the Executive Director for the National Invasive Species Council, to set the context. So just let me sort of give that mouse control to Stas and hand it over to you, Stas.
Great, and I will presume that I've got that now. Good afternoon or good morning for some of you, depending on what time zone you're in. My name is Staj Burgel, and I'm the Executive Director for the National Invasive Species Council. And as Elizabeth kind of noted, we've got a bit of a whirlwind tour presentation for you on some of the federal activities that are ongoing with regards to environmental DNA. So Jeff and I are going to start at the National Invasive Species Council level with some of the work that we've got going on. And then we're going to hear from Adam and Yael more specifically about what they are doing within U.S. Geological Survey as well as Bureau of Reclamation. Now, if I can advance the slides. Jeff, I don't know if you can advance the slide for me. There we go. So quickly, as I mentioned, I'm going to start with just a little bit of context about how NISC has gotten into the discussion on eDNA, and then Jeff is going to go more into the particulars there, and then we'll hear from Adam and Yale with regards to the work of USGS and BOR. And one of the interesting sort of things to note about this work is we're really trying to, I'm trying to get to the next slide. We're really trying to sort of complement the work that the council does at a sort of broad interdepartmental level. So we have 12 different departments as well as 4 White House offices that are members of the council. And so we take a very sort of broad whole of government approach to invasive species, really identifying the issues that cross various agencies and departments and really providing some of the leadership in terms of moving forward from the federal government as well as interacting with states and other stakeholders. And to this approach, we're interested in complementing the work that is specifically being done by the particular agencies themselves. So in that regard, it's really how do we leverage the work that they're doing and then further advance it from an interagency and interdepartmental perspective. Now, looking back, NISC has done sort of a lot of work both in sort of substantive as well as regional issues, and that's been sort of carried out by a range of different actors. We have had an advisory committee that's developed a number of substantive white papers. We have interagency federal task teams that are working on these areas as well. And then many times it's the work of the staff for the council itself. And thinking about, where does the council work potentially intersect with the work of the Western Regional Panel, with states and others in this area of eDNA, it might be useful to think about some of the other work that we've done in the past by context. For example, in the area of information management, we did a bit of work working with National Aquatic Species, the NAS, the Non-Indigenous Aquatic Species Database, with bison and other repositories for invasive species information. How do you establish interoperability? How do you mobilize data, share data across those? And then this kind of work was broadened out, for example, with the work that Western Governors Association has done with regards to data mobilization. Similarly, in the area of the building consensus process, we worked with ANSTF around looking at what federal agencies could and could not do regarding the movement of aquatic invasive species onto and off of federal lands. And that sort of became one of the building blocks in terms of the work that subsequently the agencies and the states have done with regards to recreational watercraft in the West. Get to the next slide. Over the past year or year and a half, one of the things that we've really been trying to do is to prioritize the work that we're doing and make sure that sort of capacity meets demand. In the past, with our work plans and management plans, we've had upwards of 80 different sort of thematic and strategic priorities. This time, moving into 2020, we really sort of switched and we have sort of an annual work plan. And that annual work plan this time around has 6 priorities. And so really, that helps us, one, focus our attention and our effort, both from the NISC staff and really when we're communicating with folks outside of the council, it's a lot easier to identify the key priorities that NISC and the NISC member agencies are working on. And so in this regard, environmental DNA assessment and coordination has been one of those particular activities. And I'll give the floor over to Jeff to talk a little bit more about that.
Great, thanks, Josh. And a quick sound check. Hope I'm coming through.
We hear you.
Okay, great. And apologize, I think, and just a heads up to Adam and Yale is maybe I might have to advance the slides. I will try to give you all control of those. So, moving on though, with respect to these 6 thematic priority activities that Josh has highlighted in our annual work plan, the environmental DNA was kind of given to me to lead that effort, and I think we've made some good progress over the year, and are looking to have a paper kind of from this federal perspective as to what are the considerations for invasive species managers in the use of eDNA. A lot of the publications that have come out to date have focused on that, the technical capacity of eDNA, and what we're hoping to through this group and the publication is to consider kind of that technical expertise, but geared towards an audience of invasive species land managers, or invasive species land and water managers, invasive species managers. To kind of quickly go over kind of what we're thinking of with this group is the Great Lakes as an example, and you'll see more about that in John's presentation shortly. And then kind of some examples of a federal and international work with respect to eDNA, and then kind of our outline of our preliminary guidance. So just quickly going through that, I think this, in many ways, the Great Lakes and monitoring for the Asian carp is an exemplary with respect to eDNA, implemented at a broad scale involving multiple federal agencies as well as state and local officials, and probably I think it's the longest standing application of eDNA used for invasive species coordination. And as you'll see in John's talk, it's operational, but it continues to evolve. And I think that that's sort of the part and parcel with a technology that's less than about 10 years old with respect to the capacity that continues to improve, and how do you take that improvement and operationalize a product so that you're consistent, but yet take advantage of those improvements. The other thing to call out, another thing to call out, is the National Ocean Atmospheric Administration's omics strategy that just came out earlier this year, calling for this attention to omics in general. And this is, as it notes on the slide there, referring to DNA, RNA, protein, and other techniques that get into this molecular work, and that it's worth calling out within that omic strategy that objective 2.1, which falls under goal 1, explicitly calls for invasive species. And Jeanette Davis, who is a senior advisor from NOAA to the National Invasive Species Council, has been deeply involved in that omic strategy, and is making sure that invasive species are kind of well recognized in as far as the strategy itself, which is out, and then the implementation of that strategy, which is forthcoming. We also want to recognize that there are international efforts, and members of the team have been integrated with, we're recognizing that we're coming from these international efforts and actually participating in those. And so, we certainly want to recognize that there's insight there, and we're building on that and kind of complementing and participating in this work. And several of the committee members were on the review panel for this Canadian guidance that's coming out. Members of our group were presenters at the International Workshop on Environmental Genomics. And this DNA Aquanet validation scale is a very useful sort of standard that we're looking at. So those types of considerations are being brought into what we're doing. To summarize kind of where we are and where we'll go, and this is kind of a draft figure 1 in the manuscript, and I think this highlights a little bit of where this work is different than a lot of the work that's been done. Most of the publications, or the majority of the publications, are kind of on that lower portion of this pyramid, which is on the technical tools. How do you go about utilizing eDNA? But that, and this group, and I'm sure Western Regional Panel and Adam and Yale in particular, brought to the attention of the group to consider, why should you consider using eDNA? Can it help with your invasive surveillance needs? And then what are the important operational implementations? And then you go into those technical details. So what we're trying to do is give this perspective, the technical expertise from the eDNA work within that perspective of deciding whether environmental DNA is right for you or not. And I think that's summarizes where we are with the NIST plan, and I'm going to pass it on to, I believe, Adam is next, and I'll try to give you control, Adam, of the mouse, and so hopefully you can
I'll just tell you to
begin.
do it. All right. Well, thank you. I'm attempting to represent the entire USGS here. I think it's important to point out that a recent survey that was done within the USGS identified over 300 projects that were related to omics, so something having to do with DNA or RNA based detections. So a lot of work is currently being done by the USGS. I'm going to highlight just a very small portion here, and hopefully a portion that is of interest to this audience. I think it's generally recognized that during all the rapid evolution of environmental DNA over the past 12 plus years, many of the finer, arguably more important details, kind of the quality assurance, quality control details, have been overlooked. There was a big rush to really show what eDNA could do, and much less time dedicated to really figuring out how to do it well. So the U.S. Geological Survey is actively working to fill in these current gaps and these current holes by working very closely with federal and state partners. So I'll touch upon a few things that we as an agency are doing across the country. In general, we're really trying to bolster and shore up lab protocols, field protocols, and result interpretation. In the lab, we're working very hard to identify and vet protocols that provide repeatable and reproducible results. And we are also working very hard to develop additional methods to decrease the potential for false positives, since we have come to learn this is a huge concern and often a limiting factor for operationalizing this tool. In the field, we are working with partners to vet methods that reduce contamination potential, and then also developing statistical tools to help inform the number of samples that need to be collected to minimize false negatives. And one of the other evolutions that the science body has made and the USGS has made over the last 4 or 5 years is that we recognize that eDNA sampling can provide incomplete and uncertain and confusing information. So we have been working very hard to develop decision support tools to help place eDNA results into appropriate and hopefully better context for managers. Next slide, please, Jeff. You have control? If I do. Here we go. So in addition to bolstering these methods, yeah, one too many. There we go. In addition to bolstering these methods, we are working with partners to apply these tools. And again, there are over 300 projects. I've just selected a couple that are very informative and applicable. We have examples of eDNA monitoring programs that are used to not only monitor invasives, but also rare native species in national park lands across the country, such as the Grand Canyon area. We're also using eDNA sampling of gut contents of invasive predators to try to gain more biological information about what impacts non-native invasive predators could be having. We're using these eDNA tools to monitor entire communities from plants all the way up to fish. And we are also using eDNA to evaluate the progress of eradication and control efforts. Those are just a couple of examples of how we're using the tool and how we're studying the use of the tool. And as far as early detection goes, we have programs underway that are using both human and robots to sample eDNA of Dracaena mussels across stream gauges, specifically in the Columbia River Basin. We're developing tools to sample not just for these, invertebrate and invertebrate invasives, but also for diseases in both fish and wildlife. And we are also working very hard to develop portable on-site eDNA detection kits so we can have some real-time information versus the days to weeks lag time before you get results back from the lab. And I think most importantly, and maybe the biggest advancement of all this, is that we have been working very closely with managers and technical experts from both universities and other agencies to develop minimum data standards for adding eDNA data from these early detection programs into the USGS non-Indigenous aquatic species database. So in 2021, so really in just a few months, we hope that eDNA data will be able to be viewed alongside traditional specimen data. And we think this is a really important step because these minimum standards really should help to guide better use of the tool at both the field and lab level. So we're really excited about this development. Thank you. Yeah.
Hey there. Thank you, Adam. If I can move on to the next slide. So at Reclamation, we have been continuing our sampling for early detection and monitoring of presented mussels. And in fact, both using eDNA and traditional microscopy, we've been expanding our eDNA sampling and detection in partnership with some of the WRP members, in fact. And I know this year got off to a rocky start for a lot of people in terms of their field season. At state, though, we've actually collected or received samples from 157 water bodies from 14 states in the Western Regional Panel and Reclamation territories. And we're in fact very close to being on par with our normal sampling load and sample analysis, which we were not expecting a few months ago. In addition to that, many of you may be aware that we've been working on updating our standard operating procedures for our field sampling, particularly in regards to methods for sample preservation to ensure sample integrity and robust analysis, as well as associated updates to sample and shipment labeling. We've also been continuing a variety of research projects to further optimize eDNA methods, as well as to work on development of new approaches, which I hope to present more on in the future. And finally, in the last few months, Reclamation has been working on updating the invasive mussel research roadmap, which we use to guide funding through our Office of Science and Technology, and that includes guidance for future work on environmental DNA. So I'd like to thank the leadership team for, see if I can move on to the next slide. Jeff, there we go. I'd like to thank the leadership team at NISC, Stosh, Jeff, and Kelsey Brantley, who've done a remarkable job of organizing and leading this group, as well as all of the other members of the eDNA task team. It includes a remarkable collection of experts from across federal agencies. So thank you for your attention. We'll be very happy to answer any questions at the end of the session.
And I think that takes care of our whirlwind tour, and we didn't even pay Yale to say that, so thanks, Yale, that's generous. And it is quite an amazing team that we've got connected through this. So I think we're right at about 20 minutes, so I guess we'll hand it over and watch for questions on the other in the half-hour block. And appreciate the Western Regional Panel for letting us share this information with you.
Oh, you might be muted there, Elizabeth. Yep, we're not hearing you. Nope, no audio. Yeah, you want me to keep moving? All right. So next up, we have John Amberg, who is doing a presentation called Building Confidence in eDNA, Asian Carp and Beyond. John is a supervisory biologist at the USGS Upper Midwest Environmental Sciences Center in La Crosse, Wisconsin. He leads a team of scientists that integrate molecular techniques into natural resource management with a primary focus on aquatic invasive species, early detection, rapid response, and control. He played an important role in the development of the eDNA monitoring program for Asian carp and continues to provide assistance for that program. His group has also developed a portable DNA detector now being used by natural resource law enforcement to monitor for the presence of aquatic invaders in bait shipments and other potential sources for organism introduction.
All right, thank you. Hopefully everybody can see my screen. So I want to thank everyone for this opportunity to talk about our work with eDNA and monitoring Asian carps and how we're working to build confidence in the method. I want to start by acknowledging the U.S. Fish and Wildlife Service, particularly the Midwest Fisheries Center and their Whitney Genetics Lab here in La Crosse, Wisconsin. They are the ones who implement the Asian carp eDNA monitoring program and were instrumental in the development of the program. So I want to give you a little bit of, give you an outline of my talk. So I'm going to start off by giving you a brief history lesson, kind of with the focus primarily on how everything started off with eDNA and Asian carp, and then how we're working to build the science and understand the strengths and weaknesses of eDNA for Asian carp. Then kind of switch to how it became operationalized, and then kind of end with some of the things that we're doing in the ways to improve it. So continuing advancements. So everything kind of started off back in the late aughts, about 15 years ago. A group of scientists decided to try a method out that was some folks in France used to detect frogs by collecting a water sample and then extracting the DNA in that water sample. They wanted to see if they could detect silver carp and bighead carp in the Chicago area waterway, and that's this section through here. You know, they collected water from sites that had really high numbers of fish, and then sites that shouldn't have any fish, particularly sites above the electric dispersal barrier in Chicago. This electric barrier is what separates the Great Lakes basin from the Mississippi basin. So it protects fish, protects the Great Lakes from fish moving from the Mississippi into the Great Lakes and then vice versa. In the results, what they ended up finding is they detected the DNA of silvers and bigheads where the fish were, but they also got a detection on the Great Lakes side of the basin. So this kind of made resource managers scramble and get nets set into the water and try to capture the fish. And one thing to keep in mind is that traditionally eDNA takes days, if not weeks, and sometimes months before you get a result. So the likelihood of them actually capturing a fish in that location is pretty slim. And so they didn't capture any fish. So what they ended up doing is using rotenone, the pesticide, to kill all the fish within the system. Once that huge operation finished, they resulted in no dead fish of the target species. Lots of dead fish, but none of the species that they were looking for. So this right off the bat caused some skepticism on the utility of eDNA. This is when we began kind of the eDNA calibration study, which was led by the U.S. Army Corps of Engineers in collaboration with the U.S. Fish and Wildlife Service and USGS. The one of the first goals was to identify new markers for detection. The original markers worked, but it was a proof of concept study is what the scientists did. And they weren't the best markers, and they definitely could be improved on. So you can see we actually screened through a wide range of markers and then identified a suite of markers that actually would be suitable. What we didn't want is we don't want just a single marker. We wanted to have multiple markers for use that would target different sections of the mitochondrial genome. And this is so that we would have some sort of validation built within the assay. So if you get a positive detection with one would expect that you get a positive detection with the other marker as well. We also wanted to find a way to sample. The idea is for rapid response, early detection, rapid response, and having months of time in the lab or processing doesn't lend itself very well to rapid response. So we ended up identifying a method, and rather than filtering water that's coming out of the Chicago waterway, which is incredibly turbid and can take hours to filter a single sample, we developed a method to use centrifuging the sample and pour off the water and extract the DNA from the pellet, which is now what the U.S. Fish and Wildlife Service is primarily doing. And then lastly, we wanted to also determine whether or not DNA could enter into the system through some other mechanism rather than a fish swimming in. So one of the concerns in the Chicago waterway was birds that could be potentially transporting it, eating fish in areas where the fish are really abundant, and then excreting that DNA into areas that don't have fish. And the other concern was on barges, and this was a major concern because barges are moving from infested areas where they're encountering the fish and then moving it above the electric barrier. So a large chance that they could potentially be transporting. And in both cases, we identified both fish or birds and barges were capable of transporting some of that DNA. And that all got wrapped into kind of the way that you would actually sample. So you'd avoid going into barge slips. You would avoid sampling around rookeries. All of this kind of got compiled into a report and also a quality assurance project plan that the U.S. Fish and Wildlife Service uses. This is a pretty detailed overview of the entire process that they use and makes things really quite transparent. So if you're interested in what they're doing, it's outlined in this. I think it's 160 some pages long, maybe even longer than that now. And really, one thing that this does is it takes out that it makes it really transparent, and it takes away that big scary black box that genetics seems to have with it. So the one thing that's also included in the quality assurance project plan, or the QAPP, is a communications plan. And this is really important. This is actually a plan if a sample comes back positive that you're not suspecting it to be positive. What ends up happening is you can follow that procedure, and without it, you end up freaking out a large number of managers. So it's really great to have that plan just in case you do end up with a positive detection. One thing, all of this is all conducted by the U.S. Fish and Wildlife Service, and it includes everything from kind of organizing and getting the samples to actually reporting the results. One thing to note about the QAPP, though, is that it's actually a living document. There's always an opportunity for improvement. Nothing is really perfect. So there's always time, there's always questions that we have. So things like, where and when to sample, how many samples do we actually need to have any certainty in detection. What are some of the other pathways that organisms could be introduced into the system? And then rather than forcing eDNA down the throats of people, is can we integrate some of these techniques with some traditional techniques? And we're doing that right now, and I'll talk about that here in a little bit. But incorporating things like high throughput sequencing to get at a more broader detection potentially. So we want to start off with the question of where and when to sample. We use the Wabash River since it has a bunch of tagged fish already in it. There's I think over 300 fish that are tagged, and we know where they're at throughout the year. We sampled 24 different locations across the river at 3 different sites along the river every 10 days for about a year. We found that you don't want to sample in the middle of the river. What you actually want to do is sample more towards the edge of the river. This is something that we typically would do, but we wanted to put data in it so that we knew when we modified the QAPP, we actually supported it with data. And this was something that we knew, and now we had the data to be able to demonstrate that. One thing that was interesting is that it's not universal. So it changes, that location will change depending on flow rates and the location that you're actually sampling along the river. So along straight stretches is going to be different than along bends. The other thing is that to get increased detection, we were able to detect fish throughout the year, but it changed throughout the year as well. So in sometimes you had a lot greater probability of detecting a fish, whereas other times it became a little bit more difficult. The time if you're going to go out and sample for Asian carp, you really want to get a high detection probability, you would sample during a spawning period. So the fish seem to be shedding a lot more DNA during that time. And those are all things that the Fish and Wildlife Service now uses within the QAPP for their monitoring. So another area is we wanted to decrease uncertainty. One thing about working with eDNA is you have certainty upon uncertainty. So you have to ask yourself the question of, is the DNA actually present at the location? So DNA isn't like a chemical that's mixed with water. Instead, it acts a lot like an organism, and it aggregates or accumulates in particular areas. So you need to be able to find those areas and understand how that DNA is acting within that system. There's uncertainty on whether or not you actually captured that DNA when you go and collect that water sample. But you also have uncertainty on whether or not when you extract the DNA from that sample, whether or not you in fact extracted the DNA. One thing that might be a surprise is that DNA extraction is not 100%. In some cases, it's much below 100%. So you're only going to extract a portion of that DNA probably out. So you want to know there's added uncertainty into that. And then when you end up with an aliquot from your sample, you might end up with 50 microliters or 100 microliters. And the question is whether or not you're going to be able to detect or transfer some of that DNA from that sample into one of your replicates. So we wanted to know, was it better to sample a particular site on a river multiple times, or is it better to collect fewer samples and run your analysis on that one sample or 2 samples more frequently? And what we ended up finding out is that it seems to be about a balance. It's a pretty good balance between the two. For eDNA with Asian carp, we end up having to collect about 50 to 100 samples at a location. And then That's if you're going to be running about 4 to 8 replicates per sample. With 8 replicates, you're going to get a lot better probability of detection. With 50 samples, that's the black line here, with the 8 replicates. That is what the U.S. Fish and Wildlife Service uses. We also wanted to consider alternative pathways. Adam alluded to it earlier about a potential point of use or a portable DNA detector. You know, one way that fish could potentially get into the Great Lakes, so there was a study that was done back in several years ago where they looked at the different bait shops and actually found Asian carp DNA in the bait shops. So there was concern whether or not the bait trade was potentially a source of introduction. So we actually started to come up with ways to be able to test this. Unfortunately, the standard methods for eDNA are a little bit too time consuming. You tend to want to have something that's going to be near real time, and also something that's going to be really quite simple for a user to use. And then ultimately provide those results within a minute. So we actually took a page out of the medical field and developed a point of use kit for Asian carp. This uses an enzyme that's isothermal, and it provides results within about 30 minutes of collecting the sample and processing it. And it's really quite simple to use. You don't need to be a geneticist to run the test. And I'll kind of show you some videos here next on if they work. So this is a law enforcement officer that is collecting a sample. So there he's putting a filter on an inline filter. It's just attached to a peristaltic pump where he's going to attach a cordless drill to run the pump. And that is a bait transport tank that has, not sure exactly which tank this is, but it would either have 100 or 10 silver carp mixed with 30,000 fathead minnows, or 1 silver carp mixed in with the 30,000 fathead minnows. So you just run out. He collected 500 mils of water through that filter. Just puts the filter into a conical tube and ready for the next set of processing. It doesn't take much longer than that. So the next video is kind of the next phases. So this is another officer who's kind of setting up the reaction. And this is the reader that's in the background, really quite simple. So that's the sample that he had put. There's a lysis buffer that you pour over the filter that basically lyses all of the cells that are on there. And then you fill up a vial. You then will take that vial and actually fill the tubes that you see down here. These are controls that she has as well. So she'll fill those to a certain level. That's a pipette that is disposable that will collect 40 microliters of a sample. So then you just put it on the machine, mix it and put it on the machine, and then run. So the big question is whether or not it works, and it did. It works actually quite well. We were able to detect a single carp in one of those chambers with pretty good certainty. If you have more fish in that you're looking for, you definitely you got a lot better chance of detecting them. One thing to note is that this excludes the 15-minute collection. That 15 minutes is 15 minutes after the carp was put in there. So this is 30 minutes and beyond. So the fish wasn't swimming around in the tank for very long. So again, it worked. One of the things that I think is really great about this tool is that it doesn't take a molecular biologist or a geneticist to actually run the assay. Instead, you could be a novice. It took about 15 minutes to train the person, and those who have no background in genetics or molecular biology are able to run results. There's no statistical difference between the experts and the novices. So one of the other things that we're doing is integrating some of these traditional, integrating these molecular tools with traditional techniques. And it's kind of like with the point of use care. The point of use isn't a definitive test. Don't think of it in the terms of it being a genetic test like they would use on the TV show CSI or something like that, and saying that, yeah, you got it. It's you know, your DNA is at the crime scene, so you committed the crime. Instead, your DNA is on a cup, so it might investigate it a little bit further, and that's what the law enforcement is using it for. So we're integrating it with new tools. There's one thing that we're really kind of working on is trying to improve the efficiency of what we refer to as ichthyoplankton tows. So fisheries biologists will go out and collect water samples and through plankton tows, and then screen through those plankton tows. That was not what I wanted to do. Let me go. Are you able to see my screen still?
I can see a slide. Yep.
You can see my slide. I can't see my. There it is. All right. So I just started looking at my face, which I don't really want to do. So you can imagine screening through these, going through these plankton tows where you're trying to find an egg or a larvae, and this could take weeks, if not months, to screen through, you know, thousands of samples that look like this. So we thought of as, hey, instead of actually doing this, they're preserving that. Can we actually use a method, a molecular method, to screen those, not make it so that it's definitive, but instead prioritize which samples that you're going to actually pick first. So the idea there is, can you actually, is there more DNA of the targeted organism? If there's more DNA, there's more like a better chance of an animal being in there. So let's start with those samples that are most likely going to have an organism in there. Because if you're looking for an invader early on, you'd want to get to it right off the bat. So running these tests, and we only used a mil of liquid and of the preservation fluid, and it actually worked really, really quite well. In fact, in all cases, it was able to kind of predict which organisms, which samples had the best chance of getting a detection. So it prioritizes the picking. Now we're starting to move into, we did this with a targeted approach. We're now trying to incorporate high throughput sequencing so that you're not just looking for one species, but we'd be able to look for many species. So we'd be able to build a invasive library and then a suite of markers, and then screen through those within using high throughput sequencing, and be able to detect if there's anything potentially in there, so that the folks that are picking them will in fact have a fish in hand. So I'll summarize by saying that you know, we had our ups and downs with eDNA and Asian carp, but we addressed those initial concerns with the calibration study, which I think is really important regardless of what species you're working with. We've been able to actually get it operationalized and have a quality assurance project plan established. And I think that's really important to have. But there's always improvements, and this is kind of like, and it has been a lot like building a plane while it's actually in flight. There's always some rooms for improvement. we have been successful in increasing the confidence in detecting the DNA, but there's still remain some challenges in with linking a live fish with eDNA. And I think that can be overcome by integrating this type of technology with traditional techniques, things that people are more comfortable with. You know, one thing to consider is eDNA has been around for about 15 years now, but there's definitely improvements that can be done, but they're still improving and doing methods development on electrofishing, which came out back in the 1950s. So they have 60 years on us. So incorporating this with some of these traditional techniques, I think would be hugely beneficial in getting acceptance into management. And then really, I think that the last point that I want to make is that all of the work that we've done with Asian carp is really quite transferable. So you can transfer the same approach to really any species that's out there, and follow that same procedure. And with that, I'll end and answer any questions, I think, at the end. So now let me see how I can stop showing my screen and.
Okay, thank you. Can everybody hear me now?
We can hear you.
Oh, wonderful. Sorry about that before, and thanks, Mason, for stepping in there. Great. Well, thank you so much, John. Next up, we have Patrick Youthy from Montana Fish, Wildlife, and Parks. We're going to switch gears into learning about our fish. Patrick received a Bachelor's of Science in Fisheries and Wildlife from Frostburg State University and an MS in Fish and Wildlife Management from Montana State University. After grad school, he worked for Idaho Fish and Game, monitoring salmon and steelhead response to habitat restoration efforts. He currently works for Montana Fish, Wildlife, and Parks in Missoula, where he is the Blackfoot River Fisheries Management Biologist. And the title of today's presentation is Fisheries Conservation in the Scapegoat Wilderness, Securing a Native Trout Stronghold in the North Fork of the Blackfoot River. Thank you for being here, Patrick. Take it away.
Thanks, Elizabeth. Thanks everybody for tuning in. Today I really wanted to share this kind of long-term project that we're finally hoping to implement next summer. This will definitely be more of a fisheries management talk, but I hope it's relevant to many of you in the audience given its non-native species context. Before I dive in, I definitely want to thank my FWP co-authors. I've been involved with this project for only 2 years, and a lot of these folks listed here have been on the project since inception. Definitely want to thank Pat Clancy, who helped me develop this particular presentation for one that we gave about a month ago during a public meeting regarding the project. I also want to thank our Forest Service partners. This is definitely an interagency collaborative project, and they've been really instrumental in helping us craft this proposal to fall within the guidelines of working within wilderness. So there's a lot of challenges with this particular project, and they've been immensely helpful in helping us get to the point that we're at now. So West Slope cutthroat trout have the largest geographic distribution of all the 14 cutthroat trout subspecies. And here in Montana, they were broadly distributed throughout the upper Missouri drainage, as well as all of the drainages on the western side of the divide, on the west side of the state. In total, they occupied about 29,000 miles of habitat historically. And when we look at their range within Montana today, conservation populations, those that have less than 10% hybridization, they're only occupying about 16% of this historical range. And then looking at our intact, genetically unaltered populations, they occupy just over 10% of the historical range. So a significant reduction in distribution. And Much of this has been attributed to legacy effects of land use that has resulted in significant habitat degradation, as well as the historical overharvest of the subspecies. Non-native species have been implicated in significant declines, and in the context of cutthroat trout, rainbow trout are especially problematic because they readily hybridize with cutthroat trout in waters where they co-occur. And more of an emerging threat, as well as one that's anticipated to pose one of the greatest challenges to long-term persistence, is climate change. And interestingly, there's been a lot of recent research kind of looking at the synergy between non-native species effects as well as climate change, and how those abiotic factors associated with climate change are essentially driving further hybridization as well as increasing the distribution of rainbow trout in these waters where they're sympatric. So in Montana, in addition to doing targeted habitat restoration as well as restricting harvest in waters with natural reproducing populations, a really common as well as one of the most effective techniques for conservation of the subspecies has been either the intentional isolation of conservation populations or taking advantage of habitat that's been isolated and then restoring the genetic status of those populations above those barriers. So looking up in the South Fork of the Flathead River in northwest Montana, Hungry Horse Dam isolated a significant portion of the South Fork Flathead drainage, which harbored a genetically unaltered population of West Slope cutthroat trout. Throughout the 1900s, non-native trout were stocked in many of the headwater lakes. So a project that just wrapped up a few years involved removing those non-native fish from headwater lakes, primarily with rotenone, in order to prevent further hybridization and then deal with the existing hybrids in that basin to maintain the conservation status of this secure isolated population. Looking at Silverbow Creek in the upper Clark Fork drainage, this was a man-made barrier erected specifically to isolate the genetically pure cutthroat trout that inhabited the upper portion of this drainage above this location. Similarly, in the West Fork Bitterroot irrigation reservoir forming Painted Rocks Dam isolated a genetically unaltered West Slope cutthroat trout population. And a project that just wrapped up last year involved removing the sole source of non-native trout in the drainage. The small tributary had Yellowstone cutthroat trout above a barrier, and after 3 years of rotenone application, eradicated that source of non-natives and prevented the future risk of spread throughout that secure isolated population. And then Cherry Creek, which has a lot of similarities to the North Fork project that I'll be discussing today, involved removing non-native fish above a natural barrier where they had been stocked, and then establishing a pure West Slope cutthroat trout population following eradication of the non-native species. And here in yellow, I've highlighted this North Fork Blackfoot project area boundary to provide some geographic as well as spatial context. And one thing I do want to highlight is that there is precedent for this type of project, but it's generally pretty rare that all these different factors align to allow for a project of this size to be implemented and have a high chance of success. So the fact that we have this in the North Fork is a major driver for why we've been focused on implementing this project for well over a decade now. So the North Fork Blackfoot, in general, the North Fork Blackfoot is kind of the crown jewel in the Blackfoot River drainage. It contains a robust population of migratory West Slope cutthroat trout and also harbors the most robust migratory population of threatened bull trout in the Blackfoot, as well as within the upper Clark Fork recovery unit. So it has considerable conservation value as well as recreational and cultural value. And the project area specifically is in the headwaters of the North Fork Blackfoot, and at the downstream end of the project boundary is this natural 50-foot waterfall, which is an impassable fish barrier. And above this, we have a pretty expansive amount of drainage area comprised of a lot of cold, clean, connected habitat and complex habitat, including 4 lakes in the upper end of the East Fork of the North Fork drainage. And it's also worth pointing out that this is entirely within the Scapegoat Wilderness. So not only does that present some logistical challenges for implementing the project, but it's one of the main reasons that we're pursuing it, is because it does contain that high quality habitat free of anthropogenic disturbance as a result of being in wilderness, and it's anticipated to be that way, hopefully in perpetuity. So this project has been in development for a significant amount of time, and that's allowed us to get sufficient baseline data to help inform and refine our project proposal to hopefully the highest chance of success. Really in the early 2000s, it started out primarily as just backcountry fisheries investigations. Not a lot was known quantitatively about what was up in this part of the remote part of the drainage. So started out with lake surveys, morphed into electrofishing surveys, and then more recently we've been relying heavily on eDNA surveys to not only corroborate some of the electrofishing data, but then also to increase the spatial scale of our sampling and go into a lot of these remote areas where electrofishing is it's challenging to get the sampling equipment in there, as well as really brushy, lots of woody debris in this burned area, and electrofishing hasn't been the most effective means of determining fish presence and absence throughout portions of the project area. We've also done habitat surveys, deployed temperature loggers to understand thermal suitability, all with the goal of informing this project. And in about 2014 is really where this project in its current form was first conceptualized. All these other projects were either ongoing or had just wrapped up around the state, and it presented a great opportunity for the department to pursue funding to do something similar in the North Fork Blackfoot. And then in 2016, the Forest Service secured some additional funding to facilitate the project. And in 2018, we did a pilot level bioassay, and I'll discuss some of those results momentarily. And then in the last few years, we've ramped up our MIPA and NEPA processes with the goals of shooting for target implementation next August. So throughout all of our electrofishing surveys, we've collected genetics data at all the sites where we've captured fish. This particular map is showing the spatial distribution of our survey sites. In yellow, those areas where we captured fish, and then in red, the sites where we did not capture fish with electrofishing equipment. So as I mentioned recently, and also just a few weeks ago, we were back there using eDNA exclusively, going to those sites that we failed to capture fish with electrofishing, determining whether fish were present or absent at those locations, and then also spending a lot of time bushwhacking into kind of uncharted portions of the drainage, headwater areas of a lot of these tributaries, just to get eyes on the ground. And along the way, we stumbled upon some presumed passage barriers, and then using eDNA to collect samples above those locations to determine whether in fact or not those really are passage barriers, all with the goals of really trying to refine our fish distribution model, understanding where fish are present in this project area. Also, we've spent a lot of time digging through historical fish, wildlife, and park stocking records, as well as newspapers from the early 1900s describing stocking events from various sportsmen's groups at the time. And what we know is that the drainage was widely stocked throughout the early and middle portions of the 1900s, as well as most recently as 1989. Most of the early records reference stocking of undifferentiated cutthroat trout, and we now know were mostly Yellowstone cutthroat trout, so not native to the Blackfoot drainage. Rainbow trout were also widely stocked, and then we do have some records of pure West Slope cutthroat trout being stocked in some of the later years. And interestingly, talking to some of the old-time outfitters, they describe that it was common practice throughout the 1940s and 50s for a lot of the outfitters to just translocate fish from below the falls up into the project area. And, you know, we're assuming that those were cutthroat or West Slope cutthroat trout. So all that information has been distilled down to our current knowledge that 19 of these streams above the falls support fish populations, as well as 3 of the 4 lakes in the project area. So I wanted to zoom out a little bit and kind of put it in the context of the broader Blackfoot drainage. And this is kind of a busy figure, but I just want to highlight a few things. The closed red circles depict those sampling locations where we have genetically unaltered West Slope cutthroat trout. And then the combinations of either black and green or black and yellow represent those locations with various hybrid forms of cutthroat trout. And then the closed black circles as well. And what we see is that the middle and upper Blackfoot drainage is really our core stronghold for pure and or conservation populations of West Slope cutthroat trout. And the North Fork in particular, I mean, it does have some hybridization occurring throughout the lower and middle reaches downstream of the waterfall, but for the most part, its hybridization is fairly limited. And then we look at the project area that's outlined here, and it really sticks out like a sore thumb. We have this headwater, large portion of the drainage that is comprised entirely of hybrid individuals. And the composition is predominantly rainbow cutthroat trout, followed by Yellowstone cutthroat trout. And West Slope cutthroat trout admixture represents only between 0 and a max of 17% throughout all of our genetic sampling up there. So it's really problematic from that standpoint. And it essentially kind of detracts from a lot of our other conservation objectives throughout the broader North Fork Blackfoot drainage. And I guess before I move on, I should mention that, you know, through the stocking record research as well as the genetic analyses, we don't have any sort of unequivocal evidence regarding historical fish presence. But regardless, we know that fish are present there now, and the fish that are there now are not what we'd expect for a natural condition or what we'd expect with a purely native population. You know, I mentioned climate change, and looking at all of our temperature logger data, it indicates that this area is, not surprisingly, highly suitable. Some portions are actually, you know, below what we would consider optimal. And when we look at some of the Forest Service Climate Shield products, it really kind of drives home the point of just how important this project area is from a long-term conservation perspective. You know, comparing 1980 and current thermal suitability to predicted mid-century thermal suitability, we see a significant contraction in the amount of predicted thermally suitable habitat. Even looking at this lower and middle part of the North Fork, where it currently supports some of our best spawning and rearing habitat for West Slope cutthroat trout, mid-century, that's predicted to be likely too warm, whereas this portion above the falls in the project area is anticipated to remain highly suitable. So further providing this impetus for you know, directing the resources to you know, implement a project of this scale to have a lasting conservation benefit. So all this leads down to our proposed action, which we are proposing to apply rotenone to approximately 67 miles of stream habitat above the falls, as well as these 3 fish-bearing lakes. And, you know, it's a little different than what is typically done with rotenone projects in Montana, where we have, you know, 3, maybe 4 years of treatments to ensure full eradication. We're proposing a single treatment followed by subsequent genetic swamping to achieve our project objectives. So the primary objective here is to establish a conservation population of West Slope cutthroat trout above the falls, so meaning less than 10% hybridization. Given the wilderness context of the project, a multiple year treatment was a non-starter. So we had to really kind of take a step back and look at all the tools in the toolbox as to, how could we at least do something up here to move the needle in the right direction. And we thought we could still have a really high chance of success with doing the single year treatment followed by genetic swamping. Typically, multiple year treatments, you're expending the same amount of effort with diminishing returns each year. Looking at the Soda Butte Creek project outside of Yellowstone, really complex habitat, provides a really interesting case study because they actually had to salvage their dead fish after each treatment. They killed about 99% of the fish following the first treatment, and then you're expending the same amount of effort the following year for the final 1%. So with the wilderness context here, we thought the level of trammeling would not be warranted for that subsequent 1%, and we thought that genetic swamping was an effective means of getting us to our project objectives. So for implementation, we're looking at doing drip stations in the tributaries and Manstem River sections, as well as treating the lakes with raft operated battery operated pumps, so circulating that rotenone laden water to assure effective treatment throughout the lake systems. And I should back up, we did a preliminary bioassay to determine our effective concentrations of rotenone, and with that we've been able to determine the amount of rotenone we'll need to bring back there, as well as how far apart we can space our drip stations and maintain an effective treatment through the project area. We intend to deactivate with the potassium permanganate, and this achieves full deactivation with 30 minutes of contact time. So given that we have bull trout and cutthroat trout right up to the falls, we're being extremely conservative in placing our deactivation station a minimum of 30-minute flow time above the falls, so that the water is fully neutralized before it leaves the project area. In the interest of time, I'll skip through this, discussing some of the logistical challenges. We'll be having to do a lot of beaver dam notching to ensure effective treatment through the stream sections. But I wanted to end on our stocking proposal. Luckily, the South Fork Flathead provides a lot of great case study and baseline information for the genetic swamping technique. So we calculated our stocking rates based upon a lot of the evaluation studies coming out of the South Fork Flathead to ensure that, based on the densities we have now, the anticipated effectiveness of the rotenone treatment, how many hybrid individuals may be left following that treatment, what sort of stocking level will we need. And out of that came these proposed stocking rates, and we anticipate stocking for a period of 3 years, the first of which would occur the fall, next fall, so that we would hopefully have genetically pure fish spawning in the project area of the spring after this rotenone treatment. And just want to end on some of the recent results coming out of the Cherry Creek project. It's kind of a busy figure, but what I want to highlight are the dots and error bars, which represent the abundance estimates. And from 2012 on is the post-treatment period, so that evaluation. And what we see is a really consistent response of, basically exponential population growth following the seeding of this vacant habitat after the rotenone treatment is completed. And we're hoping for and certainly anticipate a similar response in this North Fork project area. And fortunately, we have a really long-term data set of all this baseline data collection, which will enable us to have a really robust before-after comparison for evaluating project effectiveness. So if all goes according to plan, we would be implementing this project in 2021, and hopefully stay tuned for the results of that, and planning on about 10 years of post-treatment monitoring. So we're really excited to finally see this project come to fruition. And I will, I guess, be around for questions at the end of the panel.
Okay, thank you so much. That was really great. I'm gonna turn it over to our next presenter here. Just one second. There you are, but I don't see you in our presenter list here.
I already handed it over, I think. So
Oh, okay, perfect.
probably why he's not in there.
Sorry about that. Okay. All righty. Well, I'm excited to introduce our next speaker. And just a reminder to our participants, please put your questions in the question box and be sure to note who the question is for. And then Mason will lead that Q&A after this presentation. Okay, next up we have Zach Redman from the University of Anchorage, Alaska. Zach is a postdoctoral researcher in the University of Alaska Anchorage Chemistry Department. His research emphasizes the use of advanced analytical chemistry techniques to analyze contaminants in a variety of environmental matrices and determine the mechanisms by which those compounds are transported and degraded in the environment. The title of his presentation today is Evaluation of the Photochemical Dissipation of Rotenone and Degaline, Advancing the Development of Accurate Fate Models for Their Persistence in Alaskan Water Bodies. Thank you for being here, Zach. I'll let you take it away.
Thanks, Elizabeth, and thanks everyone else who stuck around for the very last presentation today. I'm an environmental chemist. I haven't really worked a lot in invasive species, so I'm happy to be here and learn from everybody else's presentations. But we're going to kind of take a U-turn here and start talking about what happens after you apply rotenone to control invasive species. Just as a start, in Alaska, northern pike are the primary aquatic invasive species that the Alaska Department of Fish and Game are concerned about. These pose an extreme economical and ecological threat to South Central Alaskan fisheries, which provide approximately 80,000 jobs annually. And these are such a threat due to their heavy predation of desirable indigenous species such as salmon and trout. We can see from this picture, it was provided to me by the Alaska Department of Fish and Game, that even a small pike is able to eat upwards of 25 to 30 salmonids. And you can imagine just how much a larger pike, like the one in the figure on the left here, could consume. Northern pike are native to Alaska, primarily in the northern and interior parts of Alaska. However, in the South Central region, they are invasive, and this is where a lot of tourism and fishing happens in the state. So it's really important that we're able to preserve the species that we have here and prevent invasive species from eliminating them. While there are some cultural controls for pike control, such as liberalized harvests or gill netting, rotenone has been found by ADFG to be the most effective means of eliminating pike from infested waterways. Unfortunately, and fortunately, I guess, rotenone is toxic to a wide range of aquatic species. This means that it can be great for controlling species other than northern pike, as both Patrick and John have pointed out in their presentations. However, it means that it will eliminate non-target aquatic species and require extensive restocking efforts. This means it's imperative that rotenone has been degraded by natural or synthetic sources, such as the addition of manganese oxides to help degrade rotenone from the water prior to the reintroduction of those native species. Which means that in order to facilitate this, resource managers have been tasked with weighing the economic and ecological threats of biological invasions versus the human and environmental health concerns that have historically gone hand in hand with pesticide applications, which is not an easy task. So in order to do that, they need to be provided monitoring and predictive tools to facilitate that job. Now, there have been some difficulties historically with rotenone monitoring. As Patrick just pointed out, there can be difficult to repeatedly travel out to remote application sites to continuously monitor. And in addition to that, the chemical analysis isn't always easy. CFT legumin is the formulation that's commonly applied in Alaska and in much of the western United States in order to dose with rotenone. That has 5% rotenone by weight generally and 5% other retinoids. The primary other retinoid would be dedulin, which is an isomer of rotenone. Now, from a chemical analysis standpoint, this can complicate things as it's because they're so chemically similar, they'll have overlapping signals and it can sometimes look like what you thought was a single rotenone peak is a peak splitting into 2 or 3 peaks. Furthermore, we found that many sample preparation methods, such as the dilute and shoot method, where you simply filter your aqueous extract and ship onto an analytical instrument, can be rather inconsistent in giving full reproducibility in your measurements. And other techniques, such as solid phase extraction, can be really expensive. They require you to transport large amounts of sample back to labs. They take more material components in the lab and also train personnel to reproducibly and accurately run these methods of analysis. So because it's so important that there's an accurate and robust and rapid sampling method, we advanced the method first developed by Sandvik et al., which is available in management of biological invasions, using Spinex extraction filters. The primary advancement we've made on top of this method was we were able to work with Thermo Fisher Scientific to access a high-resolution mass spectrometry instrument to identify what the primary other retinoids in CFT legumin were, because these could have synergistic toxicity, and if we're not monitoring those as well as rotenone, and they end up being more persistent, we could be seeing prolonged fish kills. So we actually ended up identifying 6 primary retinoids in CFT legumin by this method. Using this Spinex extraction procedure developed by Sandvik et al., with some minor changes to the solvent used during the extraction, we can get extraction recoveries of approximately 95% with RSDs less than 2% for rotenone and dedulin, and our limit of quantifications were below 0.3 parts per billion for those 2 compounds. So we're able to accurately reproducibly see very low levels with this method. This work is actually currently under peer review, so if you would like more information regarding this method or would like any of the more technical aspects of it so that you could reproduce it in your own labs, please feel free to contact me. Oops. So the analytical chemist in me wants to say that now that we have this good monitoring method, why can't we just go out after a dosing event, apply that method, and monitor the concentration over time and say, okay, rotenone persists for 60 days. Well, that's because the reality of it and what any resource manager or environmental chemist will tell you is that it's never as simple as that. If ADFG has done extensive monitoring efforts with all of its rotenone applications in South Central Alaska, and have found that half-lives and residence times have ranged between 10 and 246 days, which means that there's been this desperate need for more environmental fate data to help explain the variability of this rotenone persistence. This makes their jobs very difficult. So briefly, when we talk about environmental fate, when your friendly neighborhood resource managers go out and apply rotenone to kill some fish, rotenone will immediately begin to degrade from the environment by partitioning processes or chemodynamic processes. Essentially, it can store back to sediments or volatilization. From the literature, we know that there's really little to no volatilization of rotenone, and these partitioning processes are generally very slow. There are also degradation processes. So this includes hydrolysis, or breakdown by water, and microbial metabolism, or co-metabolism, which from literature we both know are going to be generally slow processes compared to these photochemical processes, which have been found to be the dominant processes, but are also highly variable. And that's mostly due to the complexity of the photochemical degradation mechanism, in which compounds can directly degrade, where they are absorbing light and breaking down with that energy, or degradation can be induced by these transient species, such as hydroxyl radicals, singlet oxygen, or triplet excited state DOM, that are generated by photochemical processes and then go and react with rotenone or other compounds and degrade them. So what we've done is set up a series of microcosms using different treatments to isolate each of these photochemical processes. Essentially, we bought a really fancy light bulb and we've shined light on a variety of treatments here with rotenone and deglanine in them to track how quickly they degrade under these laboratory conditions. And this is done so that we could get a handle on what are the primary processes occurring in the environment that are actually responsible for degradation of rotenone. We rated these at 15 and 20 degrees Celsius. Naturally, in much of the United States, and particularly in Alaska, the water is much colder than 15 degrees Celsius. Unfortunately, our fancy light bulb cooler couldn't get it colder than 10 degrees Celsius. And then even when I tried to run it at 10, the compressor froze over. So we were going to learn future work will definitely work on doing this at even lower temperatures, so this data can be used more widely. Essentially, what we've done here is had 7 separate treatments. For those of you who are very interested in photochemistry, I included them all here. Essentially, what we're looking at is pure water and lake water. Essentially, to get a baseline of our photochemical degradation, as well as determine what the effect of all the natural water constituents as a whole will have on degradation. Then we use a series of more targeted treatments, such as argon purging, to eliminate those reactive oxygen species, or these secondary reactive species here. We used isopropanol to specifically eliminate hydroxyl radicals, cesium chloride to promote a separate direct degradation mechanism. We used a reference standard of dissolved organic matter to see if in this natural lake water, if dissolved organic matter is what's primarily responsible for the secondary indirect processes. And we also used thermal generation of singlet oxygen via sodium molybdate hydroperoxide. So it's a lot of chemistry. I'm sorry, that's the last of it, I promise. Our key results from these microcosm studies were that deglanine is going to degrade significantly faster than rotenone. So even though deglanine might have been a blind spot in that it would have similar toxicity to rotenone and be hard to differentiate analytically, we know it's going to go away a lot faster. Half-lives ranged from 1 to 2.5 hours for deglanine, while they were more like 5 to 35 hours for rotenone under consistent elimination. When converting this to actual 24-hour solar cycle in the environment, that roughly turns into about 4 to 8 hours for deglanine and 18 to 126 hours for rotenone. We also noted from our triplet sensitized versus pure water treatments that both rotenone and deglanine rapidly degraded from excited triplet states. So this particular mechanism of direct photodegradation is important because it can be sensitive to other secondary quenching processes, which as a result makes it highly variable in the environment. In particular, we found that molecular oxygen was actually responsible for quenching triplet rotenone and would slow rotenone's degradation. So we observed that in anoxic conditions, rotenone degraded much faster. So in high latitude regions such as Alaska, or other parts of the United States where lakes freeze over. If rotenone was to persist underneath an ice sheet, if you had a late application and there was little to no sunlight intensity and cold conditions that might cause persistence, the sediments that may become anoxic over that time, or where anoxic conditions may exist, such as shallow lakes with higher biological oxygen demands, you might actually end up seeing faster degradation excuse me, of these rotenones. We also observed that photochemical generation of hydroxyl radicals in the environment by those secondary processes will be responsible for increasing the rate of rotenone degradation, but we didn't really observe that it would increase the rate of degulin degradation. This might have been because overall we didn't see a lot of variation in degulin's degradation rate, primarily because it just degraded so fast via direct processes. We also observed that singlet oxygen and other reactive oxygen species is not going to significantly contribute to rotenone degradation in the environment. While we did see a significant enhancement in its rate in the lab, it is notable that the method we used generates a very large singlet oxygen concentration that in some cases is about 10 times greater than what's been reported in the environment. So what this information tells us is that we'll see a slight enhancement at such high concentrations is realistic for the environment. This isn't going to be likely. And last, but I think the most interesting, is the discussion of dissolved organic matter. We saw that in both cases of the natural lake water and the reference dissolved organic matter material, the degradation rate of rotenone was significantly slower. Now, this can happen through both reduction of those direct photochemical processes by light shielding, as well as by radical quenching. It's also really interesting to note is that it appeared not all organic matter was the same, as we'll see significant differences between the two organic matter treatments used. And we also observed that it doesn't interact with different isomers the same way. Now, this is just preliminary investigation, but we're investigating the use of fluorescence spectroscopy to look at dissolved organic matter to see if we can correlate this with rotenone's photochemical dissipation. We saw that in the reference material, we get this signal A, which is different than the signal we see in our lake waters, which have fluorescence emission excitations at these B and C regions. We're hoping that in future work, we may be able to correlate fluorescence measurements of the water with faster or slower degradations of rotenones in the environment. We also looked at the degradation of rotenone and degulin at both 20 and 15 degrees Celsius. Like I said, we couldn't quite get the compressor to go down all the way to 10. We found that surprisingly, both rotenone and degulin degraded faster at colder temperatures. Now, this is the opposite of what has been observed in many other studies, and that may be primarily because we have isolated photochemical processes here, while many other processes such as hydrolysis or microbial metabolism, co-metabolism would be much slower under colder conditions. It could also be that alongside many colder conditions in the environment, there's ice cover, and when there's ice cover, there's less sunlight. In our conditions, at 20 and 15 degrees Celsius, there's no ice cover, obviously. So we ended up seeing faster degradation. And we think that this is probably due to, this is possibly due to slower triplet state quenching at lower temperatures. So remember that we said molecular oxygen was responsible for quenching these triplet states from which rotenone and degulin degrade. Removing the oxygen is going to stop the oxygen from eliminating that degradation path. And it would seem that lowering the temperature will also slow down that quenching process, allowing more time for rotenone and degulin excited states to degrade. We definitely need to do a bit more work looking into lower temperatures to see if this trend holds true. So in conclusion, we see that photochemical processes are definitely a major contributor to photodegradation. Temperature, dissolved oxygen concentration, and dissolved organic matter character all modulate retinoid attenuation. Temperature and dissolved oxygen are actually really easily monitored in the field with temperature loggers and dissolved oxygen concentration monitors that you can deploy and you can do this remotely. Dissolved organic matter is much more challenging, and our hope is that future work will both make the characterization of DOM a little bit more routine, as well as correlate it with the rate at which these protein and degradant residues will degrade in the environment and help increase our predictive capacity for them. Through this work, and what I wasn't able to present here was a lot of other rate constant molar activities and reaction quantum yields that are going to be really useful for computational monitoring. If you are interested in these things, all of this work is currently under peer review, but if you contact me, I'd be happy to share what I can. Additionally, like I said, future work will be determining the effects of temperature below 10 degrees Celsius, hopefully making some modifications to our experimental setup so that we can do that without everything freezing up. And further characterization of these dissolved organic matter characteristics and retinoid degradation should really be done, because this is probably going to be the most variable environmental property between application sites around the United States. With that, I'd like to acknowledge everybody who's helped on this project, particularly Patrick Tomko, who's the project PI, Jordan Kucher, Elizabeth Wisenkant, Kaylin, Josh, and Peter King, who are all students and technicians at UAA that did a lot of help with the day-to-day work and literature review on this project. Nicholas Molinaro from Thermo Fisher Scientific, who helped us gain access to that high-resolution mass spectrometer, as well as Christine Dunker and Robert Massengill from the Alaska Department of Fish and Game, who really helped me get a hold on retinoid use in Alaska. This work has been submitted to chromatography and environmental science and technology, so if you're interested, you can contact me and I can keep you informed on when it's ready. And this research was supported by funding from the NIH. Thank you for sticking around and listening this long. If you have any questions, I guess we're gonna start that right now. And here's my email if you aren't able to get any of your questions answered right now. Thank you.
All right, great, thank you. If we could just have all the presenters please bring their webcams up for the panel. All right, so it looks like the first question, which it was presented to John, but it's kind of a general question, and I think that all of our panelists and all of our presenters who do eDNA research can answer this one. So we might just go around and have a general discussion on this. So maybe start with John and then move from there with our other panelists. Can you describe some of the difficulties you've encountered in your eDNA research?
Difficulties have been generally around acceptance. The challenges have been with getting resource managers to accept some of the results. That's been by far the most challenging. In terms of developing the techniques and stuff, it's really quite straightforward.
But just to add to that, I think one of the challenges that we face is just how context or site-specific eDNA methods tend to be. We find out that a certain protocol works really well in a stream in Patrick's backyard, but then we go to a lake or to a very different system, and we have to almost start back at the pilot stage to figure out what are the best protocols and methods for being very effective. And so I think it's really hard to communicate to a manager that we're constantly still in a place of building off of this foundation, but really having to first do a lot of pilot testing before we can provide very specific guidance for how to execute.
Yeah, I guess I take it for granted that as we come into a particular area, that we'll go through all of the different things that need to be done for that particular area, which is going to be different from site to site to site. Yeah, that is a challenge. It's totally doable, and it's something that's easily overcome. But it's getting that resource manager to accept that, yeah, what worked in this river may not actually work exactly the same way in the next river. So
I think that's a challenge for us as well. We're in a little bit different circumstance where a lot of our work is early detection work, and we don't necessarily have the luxury of doing an optimization for every single site. We're getting samples from hundreds of different sites, and so we have to kind of settle on more of a one-size-fits-all method, which I think has worked well for us. In terms of acceptance, I think that in WRP, I think we've come a long way with that in the last couple of years, but it's an ongoing conversation for sure.
All right, if no one has anything else to add to that, we'll keep going here. So another question for John, is the rapid detection protocol for carp that you highlighted being used by law enforcement staff in the field now? You're muted.
So the original plan was that it would be used by law enforcement, and when we transferred the technology over to them, they initially started using it. One of the challenges that we ran into was the enzyme that's used. It's referred to as LAMP. It was copyrighted, and now the actual LAMP enzyme, or patented, all of those patents have now are coming off of it. They actually started coming off last year, which made the enzyme much more available for people. However, we were small potatoes. We were in the process of getting a contract to get large amounts of it and be able to provide kits to resource managers, but COVID hit, and if you've at all heard that there's a quick test that's out there for COVID, that's the enzyme that they use. So we competed with the medical industry for the enzyme, and pretty much everywhere that it was producing it says we're shipping it all to the medical field. So so we couldn't get the enzyme for this at all this year. So now that things are starting to settle down a little bit, production wise, I think that we're going to be able to start back to producing them. So we hit a hiccup with COVID, and not in the sense that you couldn't get out in the field. It was the fact that the enzyme was used up.
Okay, and from John Wolschlager, and this is also a general eDNA question. John says, I think there is often a disconnect between eDNA results and answers to questions needed to inform management decisions and actions. I'd be interested on hearing the presenters' perspectives on this from any or all.
I think having a plan in place for how you plan to utilize data that you might receive is the first step. It needs to be kind of an expectation of the range of data that you might get, detections, non-detections, et cetera, and what your plan is for how you're going to act on that, whether that's going to be additional sampling or some other approach after that.
And I'll chime in, and we're, from the perspective of the council, we're sort of taking a higher order approach, just trying to sensitize folks at higher levels within the agencies as to the range of issues and sensitivities, thinking about the false positives and how you interpret results. And so I think hopefully with more familiarity with that at higher levels, that will increase some of the receptivity as well as understanding in terms of how this tool can be used.
John, as you know, I've been trying to think about this question a lot the last few years. More recently, I've been diving into the structured decision-making literature to help, I guess, provide a different way of thinking about that question of results and ultimately what the manager's goals are and needs are. And I guess as I've come to learn, so much of it really comes with having very clear objectives from the get-go. So, what are you trying to accomplish with your overall early detection program? What are you trying to accomplish with eDNA? And just making sure that's very clear and transparent, and that you are actually using the correct tool, in this case, eDNA or maybe some other tool, to actually achieve your objectives. I think far too often we have tackled problems with tools. Any tool that was available, or any new and sexy tool that was available, without really giving enough time to think about the objectives and making sure that was the right tool for the objective. So I'm working with others on WP and other USGSers across the country that are on regional panels to try to come up with some structured decision-making frameworks for actually, making sure that if managers do intend to use eDNA, that they can align with the objectives. So no real answers yet, but I think moving forward, it's really about framing the question, having these communication plans, having these operation plans, and making sure that everything is well thought out a priori versus after you have a questionable positive result in hand.
You're muted there, Jeff. Yeah, we still can't hear you. Oh, you're muted there. Yeah, your audio is just not coming through. Sorry, Jeff. Well, does anybody else want to pick up while Jeff is figuring that out, or? So we have a so we have a question for you, John. I'm just gonna present this, and maybe Jeff will figure that out. So did they ever verify Asian carp in the bait supply?
So, yep. So no, they have never. We've never been able to find Asian carp within the bait supply. We've only done the tests, and when they have run the test in the field. Everything has come back as negative, so they've never been able to verify it. You know, the group that did the testing of all of the bait shops throughout the Great Lakes region, that did find positive detections, you know, they went back in and looked and they never were able to find it. So the potentially is it may have been some sort of artifact of the transport of the fish. Most of the bait that is sold in the Midwest actually comes from around Iowa and South Dakota and Mississippi, or Arkansas. So areas that have huge infestations of silver carp, and it's really difficult to tell the little guys from the bait. So it wouldn't be shocking if they did find them, but none of the tests have come back as positive.
Okay. Is your audio coming through yet, Jeff? No. So I've got another question from Elizabeth to all the eDNA folks. The communications plan that was presented is very useful. Is there a fillable template or training that could be provided for managers looking to try this and wanting that predetermined communications available for their employees, stakeholders, and decision makers? Can we train on this?
I provided a note in the comments that Fish and Wildlife Service has started some eDNA workshops that I believe are open to everyone, whether it's feds or states. And I believe due to COVID that they're all now virtual training. I have no idea if there's any communication plan specific training included. All that said, I think one of the hopes with the NIST document that Jeff is leading is that we've identified some of the really critical parts of the communication plan. I don't think we will have a template per se that comes out of that document, but we will, I think, identify what are some of the meaningful nuggets. The plan that John talked about, the QAPP plan, that's downloadable online, and I think there's a handful of other plans that, as the eDNA subcommittee working group chair, that we have collated from other eDNA experts and monitoring groups from across the country. So we do have I think 5 on hand that we can make available to anyone in WRP as examples.
Great.
Yeah, I was going to say that it would be, I don't know of any particular template, and I don't know if the eDNA workshops that Adam was alluding to that are going to be run by the Fish and Wildlife Service, if they're going to have that involved, if they're going to have that. But my first place would be to go to the QAPP, the U.S. Fish and Wildlife Service's QAPP, and take a look at what they actually have put together. You could model something off of that, just give you at least a starting point.
Great. Thanks. So we've got a question for Patrick. How many people will be involved in a project treating 67 miles of streams? Are you using pack teams? Is it one day of treatment?
So I think we're shooting to have a minimum of about 20 people. For some of the more challenging parts of the drainage, it'd be nice if we're able to get about 30 people for a number of days. I guess right now we're estimating it'll take between 2.5 to 3.5 weeks of treatment. Ultimately, that'll depend one way or another on what sort of flow year we end up having, how many drip stations we're needing to run, and then ultimately how many people we get. But working with the Forest Service, and since they'll likely have to put a closure on this area, we're trying to keep it under about 3.5 weeks with site preparations and so forth. I ended up taking the slide out as far as kind of logistical support, but as the proposal stands now, we're planning to use helicopters to fly in all of the chemicals, the rotenone, the detox agent, as well as the the equipment associated with the deactivation station. Given that we are working in wilderness, we're going to need to use pack stock to transport all other gear as well as personnel. So we'll likely be spending, probably 3 to 5 days getting everything into the project area and situated throughout before we start the actual rotenone application, and then using pack stock to move equipment from various spike locations and outfitter camps that we'll be using. But it'll be quite an endeavor, and yeah, we're hoping to finish up with the permitting processes here in the next month or two so that we can really, have 8 to 10 months to iron out all the specific logistics and scheduling personnel and rounding up volunteers if we need to fill in the gaps.
Just to test if I can be heard now.
Yep, we got you now.
Hey.
All right.
Try to go back to John Walschlager's question. I guess I was going to comment that I'm relatively new to this field as being tasked with it from the NISC staff perspective, but it's been really interesting to learn about something that, and coming into it from, sort of a newbie. This thing, eDNA, is what, about 10 years old? I mean, what science are we working with any monitoring techniques, whether it's remote sensing or drones, or even older than that, right? So what I found is quite an astute awareness on behalf of the scientists working with this of the limitations. They are aware of it, they're articulating it, they're communicating it to the extent possible. I think that the document we're working on with respect to communication to land managers is trying to get at that. But I would say that that's going to get us part of the way there. I think the open communication with the science community in lockstep and in close communication with the land management community is the trick, and that's kind of what we're going to advocate in that paper. But I would just kind of, again, go back to the awareness of this, the technical community, and I forgot to mention the government environmental DNA working group that Rick Lance leads with the Corps of Engineers, and folks are on that committee or on that group. I kind of always talking about these things and making some good progress and how that communication should unfold to avoid kind of maybe some of the lessons learned in the last 10 or so years. But again, keep in mind how new it is. So that's what strikes me, is how new it is and how aware the people involved with it from day to day are aware of those limitations.
Great. So we've got a few more minutes left. So I'm going to run one more question, and it's also to the eDNA folks. It says, will the eDNA detections reported to USGS NAS co-locate or mixed within the current detection records? If so, how will the user determine an eDNA detection versus other detections? From Nick Zerflu.
Hey, Nick, I'll take a stab at this since I presented it. This work is really being led by Wes Daniels, who you probably know, who leads the NAS group. From everything I have been made aware of, it'll be very clear when you are dealing with eDNA detection versus a detection from a traditional method, so a direct observation. So I do believe that it'll be they will be commingled, but it'll be very easy to tell when you have an eDNA versus a non-eDNA detection. And I believe, if I understood correctly, you can even kind of turn off and on the eDNA data through kind of like a filter as well, if you wanted to limit your observations. But as Jeff just mentioned, the USGS NAS group is aware of the limitations of eDNA, and they're aware of the concerns by managers of interpreting these data for decision making. So they want to be as clear and transparent as possible. And I think they're doing a really good job of doing that. And so I know that they're going to be very receptive to feedback. For example, if you think there's confusion when looking at those data, they want to know that. So I think that's one thing that they're trying to prevent. They want this to be very good, clear communication.
All right,
I'll
thank
just
you,
say
Adam.
that's my understanding as well, is they'll be very distinct. They'll be handled as separate data sets, and they'll be very clear and distinct iconography when you map out one or both, which is which.
All right, well, thank you to all of our presenters, and thank you for answering all of our questions, and thank you to everyone who attended today. I'll pass it back over to Elizabeth.
Hi, hopefully you can hear me okay. All right. Well, again, just thanks to all of our presenters today. You did an outstanding job. I learned a lot. I really appreciate your time and expertise and energy to be with us today and share this information. And thanks to Mason for jumping in and running a great Q&A for us. Tomorrow is our business meeting, and it's at the last day of our 5-day virtual conference. And I know a lot of organizations, the business meeting is not the big attraction, but I promise we have a lot of good stuff in store for you tomorrow. This is one of the most productive and engaged organizations that I have ever been a part of. And so I really encourage you, whether you're a member or a new participant, please join us tomorrow from 1 to 3 for our business meeting. And then finally, if you're a voting member, don't forget to cast your vote tonight. Check out our Facebook group and get in on the drawing that Astonish is running. And just thanks to those that joined us during the week with our coffee chats and happy hours. It was nice to see your faces. So thanks for stopping in and saying hi. And with that, everybody have a great rest of your day, and we will see you tomorrow. Thank you so much, everyone.
Yeah, thanks everyone.